Welcome to Deep Sleep Documentaries, your sanctuary for calming, immersive journeys into the natural world. Tonight, we will explore the mysterious and mesmerizing depths of our oceans, discovering hidden reefs, drifting multitudes, and the incredible life that thrives in places few have ever seen. From vibrant coral gardens to the silent abyss, each moment is designed to relax your mind and guide you gently into a state of calm. If you enjoy calming documentaries like this, you can subscribe to the channel. It helps you find these stories again whenever you need a quiet moment. And if this video brings you peace, a simple like can help others discover it too. Thank you in advance for your support. Now, let's continue into the documentary. At the surface of the world's oceans, sunlight enters the water in long, shimmering beams, slowly weakening as it travels downward. This illuminated region is known as the euphotic zone, a band of water that typically extends only about 200 meters deep. Though it represents less than 5% of the ocean's total volume, this narrow layer is responsible for producing more than half of Earth's oxygen. It is here that life begins, nourished by light, movement, and microscopic organisms that drift calmly with the currents. The key to this remarkable productivity lies in a group of organisms that are almost invisible to the naked eye: phytoplankton. These tiny photosynthetic drifters use sunlight to convert carbon dioxide into oxygen, just as trees and plants do on land. But in the vastness of the ocean, their impact is magnified. A single liter of seawater can contain tens of thousands of phytoplankton cells. Together, they form the foundation of the marine food web, supporting everything from small fish to the largest whales on the planet. Scientists estimate that phytoplankton produce between 50 and 70% of all the breathable oxygen on Earth. This means that every second or third breath humans take is thanks to life in the ocean. Satellite observations from NASA's CYFS and Aqua missions have confirmed the global scale of these microscopic forests, revealing seasonal blooms that appear like swirling strokes of turquoise and green across the surface of the sea. These blooms can stretch for hundreds of kilometers, forming living patterns shaped by winds, currents, and temperature changes. Within the euphotic zone, sunlight drives more than photosynthesis. It creates a unique gradient of energy, warmth, and nutrients. At the very top lies a thin, warm layer, known as the mixed layer, stirred constantly by wind and waves. This mixing brings nutrients upward from deeper water, fueling phytoplankton growth. Below this, the light grows dimmer, and the water cools sharply at a boundary known as the thermocline. Here, the drop in temperature forms a barrier that limits how far surface waters can mix downward, shaping the distribution of life in remarkable ways. Animals that inhabit this sunlit zone have evolved a stunning variety of adaptations. Small fish often display bright colors or shimmering scales that scatter incoming light, confusing predators. Planktonic creatures are frequently transparent, allowing sunlight to pass through their bodies and making them nearly invisible. Larger fish rely on countershading, dark backs and pale bellies that blends them into the ocean whether viewed from above or below. Even with abundant light, survival requires constant awareness and evolution. One of the most iconic inhabitants of these waters is the green sea turtle. During daylight hours, these ancient reptiles glide calmly through shallow regions where seagrass meadows thrive. Seagrasses, like phytoplankton, rely heavily on sunlight, and the turtles help maintain the health of these underwater fields by grazing on them. Studies from the NOAA Marine Turtle Research Program have shown that healthy seagrass beds support more biodiversity and store large amounts of carbon, making sea turtles an essential part of the coastal ecosystem. Another key group of creatures moving through this illuminated world are small schooling fish, such as anchovies, sardines, and mackerel. Traveling in massive synchronized groups, they use the sun's rays to their advantage. When sunlight reflects off thousands of bodies at once, it creates a flickering effect that disorients predators, making it harder for them to isolate a single target. Marine biologists refer to this as the confusion effect, an elegant example of how light can shape behavior in the ocean. But life in the euphotic zone is defined not only by sunlight, but also by the competition for nutrients. Sunlit waters are often nutrient poor, especially in tropical regions where the ocean is calm and stratified. In contrast, areas with strong coastal upwelling, such as the coasts of Peru, California, and Namibia, are among the most productive places on Earth. Here, deep, cold waters rich in nitrogen, phosphorus, and iron rise toward the surface, sparking explosive growth in phytoplankton. These nutrient surges support massive populations of fish, seabirds, and marine mammals, all connected by cycles of sunlight and motion. One of the most fascinating behaviors linked to the euphotic zone is the daily vertical migration of zooplankton. At dusk, billions of tiny animals rise toward the surface to feed on phytoplankton in the safety of darkness. Before dawn, they sink back into deeper, darker waters to avoid predators. This movement, recorded by sonar and oceanographic sensors, is considered the largest daily migration of any animals on Earth. Though individual organisms are small, their collective movement helps transport carbon from the surface to deeper layers, influencing global climate processes. Research from the Woods Hole Oceanographic Institution has shown that this vertical migration plays a vital role in the biological carbon pump. As zooplankton consume phytoplankton, they package carbon into dense particles that sink more quickly. Over time, this process helps remove carbon dioxide from the atmosphere and store it in the deep ocean, sometimes for centuries. This hidden mechanism is one of the most effective natural systems for regulating Earth's climate, silently driven by life, no larger than a grain of sand. The interplay between light, life, and motion also shapes coral reefs, which thrive only within the boundaries of the euphotic zone. Corals contain symbiotic algae called zooxanthellae, which rely on sunlight to produce energy through photosynthesis. These algae provide corals with food, while the corals offer shelter and protection. The relationship is so precise that even small changes in temperature or sunlight can cause the algae to leave, leading to coral bleaching. This delicate dependency underscores how important the sunlit zone is for maintaining global biodiversity. Even predators like sharks are shaped by the physics of light. Many shark species, such as the blacktip reef shark, patrol shallow regions during daytime hours, using the interplay of shadows and reflections to ambush prey. Their eyes contain a reflective layer called the tapetum lucidum, similar to that of cats, which enhances their ability to see in low-light conditions as they move between brighter and dimmer parts of the water column. As the sun lowers on the horizon, the illuminated world begins to transform. Colors fade, silhouettes sharpen, and the ocean's surface glows with the last traces of daylight. But even as the light diminishes, the processes it sets in motion continue. Phytoplankton drift silently, storing the energy gathered through the day. Zooplankton begin their slow ascent. Predators adjust their patterns, and the ocean prepares for the rhythms of the night. The euphotic zone is more than a habitat. It is a breathing engine of the planet, a delicate balance of physics, chemistry, and biology. Here, where sunlight first meets the sea, life flourishes in extraordinary ways, quietly, efficiently, and endlessly. This thin layer of light sustains the oceans, shapes the climate, and supports nearly every creature below. In understanding this world, we begin to understand the ocean itself, a realm where even the faintest beam of sunlight can bring life to the vast blue expanse. Coral reefs are often compared to underwater cities, places full of color, motion, and intricate structures. Yet the architects behind these vast formations are among the smallest and most fragile organisms in the sea. Coral polyps, tiny creatures no larger than a few millimeters across, work silently over centuries to build some of the most biodiverse ecosystems on Earth. Their bodies are soft, translucent, and simple in design, but through their collective effort and the chemistry of the ocean, they create limestone fortresses capable of lasting thousands of years. Coral polyps belong to the same biological group as jellyfish and sea anemones, known as the phylum Cnidaria. Each polyp has a cylindrical body with a ring of tentacles that it uses to feed and to sense its environment. Despite their delicate appearance, polyps possess specialized stinging cells called nematocysts, enabling them to capture microscopic prey drifting through the water. But most of their energy does not come from hunting. Instead, it comes from a remarkable partnership with tiny algae called zooxanthellae, which live inside their tissues. This symbiosis is the engine that drives the growth of coral reefs. Zooxanthellae use sunlight to photosynthesize, producing sugars and oxygen that the coral polyp relies on for survival. In return, the coral provides the algae with carbon dioxide and a protected space to live. Studies show that up to 90% of a coral's energy can come directly from these algae. Without them, most corals would not be able to survive in the nutrient-poor waters where reefs are found. The ability of corals to build massive structures lies in their production of calcium carbonate, also known as aragonite. Polyps absorb calcium and carbonate ions from seawater and deposit them beneath their bodies, gradually forming a hard external skeleton. This process, called calcification, occurs slowly. Most coral species grow only a few millimeters per year. Massive corals, such as brain corals, may add just 1 centimeter annually. Branching corals grow faster but remain delicate. Over decades, centuries, and even millennia, these small increments accumulate into towering colonies and wide expanses of reef. Some of the world's oldest coral formations are estimated to be more than 5,000 years old. The Great Barrier Reef, stretching over 2,300 kilometers along the coast of Australia, is one of the most famous examples. It is so large that it can be seen from space. Yet even the Great Barrier Reef is composed entirely of colonies of individual polyps, each one contributing a tiny piece to the larger structure. Researchers studying reef cores, vertical samples drilled through coral formations, have uncovered growth layers similar to tree rings, revealing detailed histories of past climate, water temperature, and ocean chemistry. Coral reefs form only in specific conditions, which makes them both unique and vulnerable. They need clear, warm, shallow water, with plenty of sunlight for their symbiotic algae. The ideal temperature range is between 23 and 29 degrees Celsius. Water that is too warm can cause the zooxanthellae to become stressed and leave the coral's tissues, a phenomenon known as coral bleaching. Without their algae, corals lose their color and their primary energy source. If stressful conditions persist, the corals can die within weeks or months. Despite these challenges, coral reefs have managed to flourish across the world's tropical oceans. The Indo-Pacific region, spanning from the Red Sea to the Central Pacific, contains the highest coral diversity, with more than 500 species. The rich variety of shapes, branching, plate-like, massive, encrusting, and delicate, creates a complex landscape with countless microhabitats. This structural diversity supports more than a quarter of all marine species, even though reefs occupy less than 1% of the ocean floor. Fish rely heavily on coral reefs for shelter, food, and reproduction. Some species hide within the branches of staghorn coral to avoid predators. Others graze on algae growing on the reef surface, helping maintain the delicate balance necessary for coral health. Small fish often use the reef as a nursery, while larger predators patrol the outskirts. The dynamic interactions among species create a living tapestry that shifts throughout the day and night. Scientific studies have shown that coral reefs also play an important role in protecting coastlines. The complex structures built by polyps act as natural breakwaters, absorbing up to 97% of the energy from incoming waves. This reduces erosion, protects mangroves and seagrass beds, and helps safeguard communities living near the shore. In many regions, reefs provide food, tourism income, and cultural significance that sustain local economies. The process of reef formation is influenced by geology as well as biology. Some reefs develop on submerged volcanic islands, gradually growing upward as the land beneath them sinks. This process was first described by Charles Darwin during his voyage on the HMS Beagle. He observed that fringing reefs near land could eventually transform into barrier reefs, and then into atolls, ring-shaped reefs surrounding a central lagoon, if the island's substrate slowly subsided. Modern oceanographic studies have confirmed Darwin's theory, revealing how corals can track sea level changes over thousands of years. However, coral polyps must constantly adapt to survive. Predators such as crown-of-thorns starfish can devastate reefs by feeding on coral tissue. Outbreaks of these starfish have caused significant declines in reef cover across the Pacific. Additionally, ocean acidification, caused by increasing levels of carbon dioxide in the atmosphere, reduces the availability of carbonate ions that corals need for calcification. Lower pH levels make it more difficult for polyps to build their skeletons, slowing growth and weakening reef structures. Despite these threats, coral polyps have evolved remarkable strategies to persist. During spawning events, which often occur once a year and are synchronized by the lunar cycle, millions of corals release eggs and sperm into the water column simultaneously. This mass spawning increases the chance of successful fertilization and allows new polyps to settle in suitable locations. Observations from the Great Barrier Reef show that these events can be so extensive that they temporarily change the color of the water. New advances in marine science provide hope for the future of coral reefs. Researchers are developing methods to grow corals in nurseries and transplant them into damaged areas. Some experiments involve selectively breeding corals that can withstand higher temperatures. Other studies focus on restoring the symbiotic algae themselves, helping corals recover more quickly from bleaching events. These efforts highlight the resilience hidden within these small but extraordinary animals. When viewed closely, a coral colony appears to be a single organism, but in reality, it is a community of individuals working together, each polyp contributing to the greater structure. Their collective growth creates habitats that support immense diversity and shape the health of the ocean. Though they are often overlooked, coral polyps are among the most influential engineers in the natural world. As currents move across the reef, they carry with them the sounds of crackling shrimp, grazing fish, and distant waves breaking on the reef crest. This constant hum of activity reveals the vitality of the underwater city. Every coral polyp, though small and fragile, plays a role in sustaining this vibrant world. Through patience, persistence, and a partnership with sunlight, they build structures that stand for centuries, reminders of the quiet power held within the ocean's tiniest architects. Coral reefs are among the most extraordinary ecosystems on Earth, supporting a quarter of all marine species despite covering less than 1% of the ocean floor. Yet these vibrant underwater worlds are now some of the most threatened. Over the past few decades, scientists have witnessed dramatic declines in coral cover caused by warming oceans, pollution, destructive fishing, and diseases that spread quickly across weakened reefs. The fragility of these ecosystems has sparked a global effort to protect, restore, and understand the forces that shape their survival. One of the most significant threats facing coral reefs today is coral bleaching, a stress response triggered primarily by rising sea temperatures. Corals live in a delicate partnership with microscopic algae known as zooplankton, which provide most of their energy through photosynthesis. When water temperatures rise even 1 or 2 degrees Celsius above normal for extended periods, this relationship breaks down. The algae are expelled from coral tissues, leaving the corals pale or completely white. Without their algae, corals struggle to survive, and prolonged bleaching can lead to widespread mortality. Scientists first documented mass coral bleaching in the early 1980s, but the frequency and severity of these events have increased dramatically. The global bleaching event between 2014 and 2017, fueled by a strong El Niño combined with long-term warming, affected over 70% of coral reefs worldwide. The Great Barrier Reef alone experienced back-to-back bleaching in 2016 and 2017, resulting in the loss of an estimated half of its shallow water corals. These events revealed how quickly reefs can decline when exposed to sustained thermal stress. High ocean temperatures are not the only cause of bleaching. Pollution from land, such as agricultural runoff containing fertilizers and sediments, can cloud the water and reduce light penetration. Excess nutrients can fuel algae blooms that smother corals. Additionally, chemical pollution and oil spills disrupt coral physiology and make them more susceptible to disease. Many reefs near densely populated coastlines show signs of chronic stress, with slower growth, reduced reproduction, and lower biodiversity. As the scale of reef decline becomes more apparent, researchers and conservationists have developed a range of strategies to protect and restore these ecosystems. One of the most promising approaches involves creating coral nurseries, underwater or land-based facilities where corals are grown under controlled conditions before being transplanted back onto damaged reefs. These nurseries allow scientists to cultivate thousands of coral fragments at a time, accelerating natural growth rates and increasing the likelihood of survival. Coral nurseries come in different forms. Floating rope nurseries suspend coral fragments in mid-water, where currents deliver nutrients and reduce the buildup of sediment. Other nurseries use metal or PVC structures anchored to the seafloor. In land-based facilities, tanks with precise temperature and light control allow researchers to monitor coral health at every stage. Programs such as the Coral Restoration Foundation in Florida and the Reef Renewal Organization in the Caribbean have successfully outplanted tens of thousands of nursery-grown corals, helping restore systems that were previously in decline. Beyond simple propagation, scientists are also exploring assisted evolution techniques to increase coral resilience. By exposing corals to slightly higher temperatures in controlled environments, researchers encourage them to develop tolerance to warmer conditions. Some programs breed corals that have survived previous bleaching events, hoping that these traits will be passed on to future generations. Others focus on the symbiotic algae themselves, identifying strains that can withstand higher temperatures or changing water chemistry. One notable initiative comes from the Australian Institute of Marine Science, where scientists are working to strengthen the Great Barrier Reef using selective breeding and heat-resistant algae. Early results suggest that some coral species can indeed be made more resilient through targeted interventions. While this approach cannot replace global climate action, It offers a potential lifeline for reefs facing increasing thermal stress. Another major focus of reef protection efforts is reducing local stresses. Even if global temperatures continue to rise, improving water quality and reducing pollution can enhance coral resilience. Many countries now implement marine protected areas where fishing, coastal development, and other human activities are restricted. Well-managed protected areas can result in faster reef recovery, with healthier fish populations and lower levels of destructive practices like anchor damage or overfishing. In some parts of the world, community involvement plays a crucial role in reef conservation. Local fishers, divers, and conservation groups work together to monitor coral health, remove invasive species, and plant nursery-grown corals. Programs in Fiji, the Philippines, and Indonesia have demonstrated that community-managed reef areas often show higher biodiversity and better recovery rates. Education and stewardship strengthen the connection between people and the reefs they depend on for food, cultural identity, and economic security. Technological innovations are also transforming how scientists study and protect coral reefs. High-resolution satellite imagery allows researchers to monitor bleaching events in near real time. Autonomous underwater vehicles, or AUVs, can map reef structures and collect data from depths that divers cannot easily reach. AI-powered image analysis helps track coral growth and identify early signs of disease. These tools provide a clearer picture of reef health and help direct restoration efforts more efficiently. One of the most challenging aspects of reef restoration is ensuring that transplanted corals survive long enough to form self-sustaining colonies. Success rates vary depending on species, location, and environmental conditions. However, even partial success can help restore biodiversity and stabilize damaged reefs. Some restoration sites have shown coral cover increasing by 20 to 40% within a few years, providing habitat for fish and other marine life to return. Coral reefs also hold scientific value beyond their ecological importance. They act as natural archives, recording changes in ocean temperature, chemistry, and sea level within their skeletons. By analyzing these growth layers, scientists can reconstruct climate patterns spanning centuries. This information helps improve climate models and deepens our understanding of long-term environmental change. Despite the progress made in conservation and restoration, the future of coral reefs remains uncertain. Climate projections suggest that if global temperatures rise more than 1.5 degrees Celsius, over 90% of reefs could be at risk of severe bleaching. Large-scale solutions depend on reducing greenhouse gas emissions, protecting coastal environments, and restoring damaged ecosystems wherever possible. The global race to save coral reefs is a race against time, requiring both local action and international cooperation. Yet there is hope within the fragility of these ecosystems. Coral polyps have survived countless natural disturbances throughout their evolutionary history, from storms to shifts in sea level. Their resilience, combined with human ingenuity, offers a chance to safeguard these underwater wonders. Every nursery-grown coral transplanted onto a damaged reef, every policy protecting coastal waters, and every community effort contributes to this global mission. Coral reefs are not just habitats. They are living reminders of the ocean's creativity and its vulnerability. They show how delicate the balance of life can be, and how essential it is to protect the natural systems that sustain our planet. The work being done today may determine whether future generations will witness the colors, sounds, and life that thrive within these fragile wonders. Through science, restoration, and global commitment, the story of coral reefs is still being written. One fragment, one polyp, and one restored reef at a time. For most of human history, coral reefs were admired from the surface, but remained largely unknown beneath the waves. Their colors, forms, and diversity were hidden behind the barrier of depth and pressure, far beyond what early divers or swimmers could safely reach. It took a series of daring expeditions, technological breakthroughs, and relentless curiosity to illuminate the hidden world of the reefs. The story of how we came to explore these underwater landscapes is one of innovation, persistence, and a profound desire to understand the ocean's most complex ecosystems. The earliest glimpses of deep marine life came in the early 20th century, a time when exploring the ocean below 30 meters was considered nearly impossible. It was during this era that American naturalist William Beebe undertook one of the most ambitious oceanographic projects ever attempted. Beebe believed that direct observation was the key to understanding life in the deep sea, and in 1930, he embarked on a groundbreaking series of dives off Bermuda, using a spherical steel submersible known as the bathysphere. Suspended by a steel cable and lowered into the ocean like a scientific pendulum, the bathysphere allowed Beebe and his engineer, Otis Barton, to descend deeper than any humans before them. Inside the bathysphere, conditions were extremely cramped. The steel sphere was barely large enough for two people to sit, and thick quartz windows were the only portals to the outside world. Yet this fragile-looking vessel carried them to a depth of 435 meters, setting a record that stood for decades. Beebe's detailed observations of deep-sea creatures, many glowing with bioluminescence, reshaped scientific understanding of life at depth. While his focus was not specifically coral reefs, his work represented a foundational moment in marine biology, the first time humans witnessed deep ocean life directly, rather than through dredging or speculation. As Beebe was exploring the ocean's vertical frontier, another revolution was unfolding: the development of more accessible underwater breathing systems. This breakthrough would transform our understanding of coral reefs by allowing researchers to move freely through their intricate structures rather than observing them from boats or submersibles. The most influential figure in this revolution was Jacques-Yves Cousteau, a French naval officer, filmmaker, and naturalist whose inventions and expeditions opened the underwater world to science and to the public. In 1943, Cousteau and engineer Émile Gagnon developed the aqualung, the first practical open-circuit scuba system. For the first time, divers could stay underwater for extended periods, exploring coral reefs at depth with unprecedented ease. Cousteau's expeditions aboard his research vessel, the Calypso, became legendary. The ship, originally a minesweeper, was transformed into a floating laboratory equipped with dive gear, underwater cameras, and small submersibles. From the 1950s through the 1970s, Cousteau and his crew documented reefs around the world, from the Red Sea to the Indian Ocean, capturing images that mesmerized global audiences. Cousteau's films, including The Silent World and World Without Sun, introduced millions of people to the beauty and complexity of coral reefs. They showed branching corals swaying in gentle currents, schools of reef fish flashing in sunlit shallows, and divers interacting with marine life in ways previously unimaginable. More importantly, Cousteau's footage revealed that reefs were living, dynamic systems, not static underwater gardens, but bustling habitats shaped by biology, geology, and time. His work laid the foundation for modern marine conservation by highlighting both the splendor of reefs and their growing vulnerability. During this same period, scientific diving emerged as a professional field. Researchers began conducting systematic surveys, documenting species distributions, and studying coral growth rates directly in their natural environments. Dive tables, underwater communication systems, and improved buoyancy equipment allowed marine scientists to work longer and more safely. As diving became widespread, the scientific community gained its first detailed maps of reef structures and their ecological processes. By the late 20th century, interest in exploring deeper and more remote reefs grew. Many coral ecosystems lie well beyond the limits of recreational scuba diving, including mesophotic reefs, those found between 30 and 150 meters deep. To reach these hidden communities, researchers turned to advanced diving technology and submersibles capable of descending safely into the twilight zone. One of the key innovations was mixed gas rebreather systems, which allowed divers to stay at depth longer without the risks associated with nitrogen narcosis and decompression sickness. Despite these advancements, many reefs remained inaccessible until the rise of modern submersibles, small research vessels designed to withstand extreme pressure, while offering clear visibility and maneuverability. Submersibles, such as the Johnson Sea Link, first launched in the 1970s, allowed scientists to observe deep reef formations and collect samples with mechanical arms. These dives produced some of the first evidence that reef-building corals exist far deeper than previously believed, thriving on steep underwater walls where sunlight still filters through the water. In recent years, a new generation of submersibles has pushed the boundaries of ocean exploration even further. The Deep-Sea Challenger, piloted by filmmaker and explorer James Cameron in 2012, reached the bottom of the Mariana Trench at nearly 11,000 meters. While that dive focused on the deep abyss rather than coral ecosystems, the technological advances it demonstrated, pressure-resistant materials, high-definition imaging systems, and automated life support controls, have influenced the design of many reef-oriented research vehicles. More directly relevant to coral exploration are the Triton submersibles, widely used in marine research and documentary filmmaking. These transparent acrylic-domed vessels give occupants a nearly unobstructed view of the underwater world, making them ideal for observing reef behavior in real time. Researchers using Triton submersibles have documented reef fish spawning events, coral feeding behaviors, and species interactions in ways impossible for divers constrained by depth and time. Another breakthrough is the rise of autonomous underwater vehicles, OVs, and remotely operated vehicles, ROVs, robotic explorers that can map, photograph, and sample reefs without risking human divers. Equipped with high-resolution cameras, sonar mapping tools, and environmental sensors, these robots can operate for hours or even days at a time. NASA's Buoy robot, originally developed for exploring icy moons, has been tested in polar waters to study cold water coral structures. Similarly, reef-focused ROVs are being used to explore mesophotic reefs in Hawaii, the Virgin Islands, and the Red Sea, revealing species and formations previously unknown to science. The data collected by these expeditions has dramatically expanded our understanding of coral ecosystems. We now know that reef-building corals have existed for more than 240 million years, surviving mass extinctions and dramatic shifts in ocean chemistry. We've learned that coral growth rates vary widely, from a few millimeters per year in massive boulder corals to more than 10 centimeters per year in fast-growing branching species. We've mapped reefs thousands of kilometers long, including the Great Barrier Reef, which stretches over 2,300 kilometers and contains around 3,000 individual reef systems. These voyages of discovery have also illuminated the threats facing coral reefs. Many deep reef surveys reveal signs of warming, bleaching, and disease, even at depths once believed to be more stable. Remote reefs, once thought pristine, show impacts from drifting plastics and microfibers. Yet the same expeditions also reveal pockets of resilience, reefs that recover rapidly after storms, coral species that tolerate higher temperatures, and ecosystems that flourish in areas protected from human pressure. Today, our exploration of coral reefs continues with greater urgency and greater sophistication. Each submersible dive, each robotic survey, and each scientific expedition adds to a growing understanding of these underwater worlds. Together, these journeys form a continuous chain of discovery, from Beebe's claustrophobic bathysphere to Cousteau's iconic Calypso, and onward to the quiet hum of modern submersibles descending into the blue. They remind us that curiosity drives progress, and that the more we understand the reefs, the better equipped we are to protect them. For millions of years, sea turtles have crossed the world's oceans following routes that remain among the most extraordinary migrations in the natural world. Their journeys span thousands of kilometers, taking them from feeding grounds to ancient nesting beaches with remarkable precision. Despite storms, shifting currents, and the vastness of the open ocean, sea turtles return to the same stretches of coastline generation after generation. The question of how they accomplish this feat has fascinated scientists for decades, and modern research reveals a story that is both elegant and astonishing. Sea turtles, it turns out, are guided not only by instinct and memory, but also by the invisible forces of the earth itself. The science of sea turtle migration began to take shape in the mid-20th century, when researchers noticed that nesting females often returned to beaches that matched their place of birth. Tagging programs conducted in Florida, Costa Rica, and Australia showed that individual turtles traveled across entire ocean basins, sometimes swimming more than 10,000 kilometers before returning to lay their eggs. Green turtles, in particular, are known for their epic migrations, journeying from South America to islands in the Atlantic, while loggerhead turtles travel between Japan, Hawaii, and the coast of Mexico. These findings led researchers to suspect that turtles possessed a navigational system far more sophisticated than simple memory or environmental cues. One of the earliest clues about this ability came from observing hatchlings. When baby turtles emerge from their nests, they make a frantic dash across the sand toward the ocean. Within minutes of entering the water, they begin swimming directly offshore, following a course that takes them into the open sea. This swim frenzy lasts for several days, and astonishingly, hatchlings head in the correct direction even when released at night or under overcast skies. Experiments conducted at the University of North Carolina suggested that hatchlings use multiple orientation cues, including the angle of breaking waves and the brightness of the open horizon. Yet these cues only guide them during their first moments of life. After that, they rely on something deeper, something embedded in their biology. The breakthrough came in the 1990s when biologist Kenneth Lohman and his team began studying the magnetic abilities of sea turtles. They discovered that turtles could detect the geomagnetic field of the Earth, a complex pattern created by the planet's molten core. This magnetic field varies across the globe, with differences in intensity and inclination that form a kind of invisible map. Lohman's experiments placed hatchlings inside specially designed coil systems that allowed scientists to manipulate magnetic fields. When exposed to magnetic conditions that mimic distant parts of the Atlantic, the turtles orient themselves in the direction they would need to swim if they were actually there. These results proved that sea turtles use magnetic navigation, a sensory ability shared by only a few species on Earth. This magnetic sense provides turtles with two critical pieces of information: latitude and longitude. Magnetic intensity varies north to south, while magnetic inclination changes depending on distance from the equator. By combining these two measurements, turtles develop a form of geomagnetic positioning, allowing them to follow routes across entire oceans. This ability is especially important for species like the loggerhead, which undertake looping migrations across the North Atlantic, riding powerful currents such as the Gulf Stream, before eventually returning to the beaches where they were born. Further studies revealed that turtles imprint on the magnetic signature of their natal beaches shortly after hatching. Each coastline possesses a unique magnetic fingerprint, defined by the local configuration of the Earth's field. Turtles memorize this signature, and decades later, when they reach adulthood, they navigate back to the same region to lay their eggs. This remarkable biological memory has been confirmed through genetic studies. Populations of turtles nesting on beaches only a few kilometers apart often show distinct genetic differences, simply because each group returns to its specific magnetic zone. The discovery of magnetic imprinting also helped explain surprising shifts in nesting locations observed in recent decades. As the Earth's magnetic field gradually drifts, a natural phenomenon known as secular variation, the magnetic signatures of coastlines change. Researchers found that when a beach's magnetic coordinates shift even slightly, the distribution of nesting turtles moves with it. In other words, turtles continue to follow the magnetic address they imprinted on as hatchlings, even if that address has migrated along the coastline over time. This has led to increases in nesting activity in some areas and declines in others, reflecting the slow but steady march of the Earth's magnetic landscape. While magnetic navigation explains the broad movements of turtles across ocean basins, their journeys are also influenced by ocean currents, food availability, and temperature gradients. Satellite tagging studies have revealed complex migratory corridors. Leatherback turtles, for example, traverse both the Atlantic and Pacific Oceans, following blooms of jellyfish that form their primary diet. Some individuals travel from the coast of California to waters near Indonesia, completing one of the longest migrations of any marine animal. Meanwhile, hawksbill turtles migrate shorter distances, often staying within coral-rich regions where sponges, their preferred food, are abundant. Modern technology has transformed our understanding of these migrations. Miniature satellite transmitters attached to turtle shells allow scientists to track movements with remarkable precision. These tags record location, dive depth, and water temperature, revealing behaviors that were once impossible to observe. They show that turtles dive to depths of over 1,200 meters, resting in cold, dark waters before returning to the surface to breathe. They also reveal how turtles respond to environmental threats, such as drifting plastics, fishing nets, and rising sea temperatures. Magnetic navigation research has also intersected with conservation efforts. Because turtles rely on stable magnetic cues to find nesting beaches, coastal developments that alter the magnetic environment, such as seawalls or large concrete structures, can disrupt navigation. Light pollution can misdirect hatchlings, pulling them toward artificial lights instead of the sea. Efforts to protect nesting beaches now include guidelines on shielding artificial lighting, restoring natural dunes, and minimizing magnetic interference. In some places, conservationists even relocate nests from eroding beaches to safer sites that closely match the original magnetic signature. One of the most remarkable findings in recent years is that sea turtles adjust their migratory behavior in response to climate variability. Studies conducted in the Pacific show that El Niño events, which alter ocean currents and temperature gradients, can significantly shift turtle migration routes. Turtles appear to sense these changes and modify their paths to avoid unfavorable conditions. This adaptability suggests resilience, but only up to a point. Rapid changes in ocean temperature and chemistry caused by human activity present a far greater challenge. The story of sea turtle migration is not just one of survival, but of extraordinary biological design. Each turtle carries within it a sensory compass connected to the Earth's core, a navigation system refined over 110 million years of evolution. Their journeys reveal an ancient partnership between life and the planet itself, a connection written not in sound or light, but in the invisible lines of a shifting magnetic field. As modern science continues to uncover the details of this navigation, the wonder of it remains unchanged. Sea turtles travel across the world, guided not by landmarks or memory, but by the Earth itself. Their migrations are a reminder of the hidden forces that shape life on our planet, and of the delicate balance that allows these ancient mariners to find their way home. The oceans are often described as the beating heart of our planet, and nowhere is this more evident than in the movement of their great currents. These powerful rivers of water flow across entire basins, connecting distant coastlines and shaping every ecosystem they touch. From the warm push of the Gulf Stream to the cold, nutrient-rich upwellings along continental margins, global currents determine where life flourishes and where it struggles. They transport heat, oxygen, minerals, and microscopic life, creating patterns that have remained largely unchanged for thousands of years. Understanding these currents reveals the hidden mechanics of the ocean's pulse, a rhythm that sustains much of the biodiversity on Earth. The Gulf Stream is one of the most powerful and well-known of these currents. Originating in the Gulf of Mexico, it flows through the Florida Straits before turning north along the eastern coast of the United States. From there, it curves eastward toward Europe, forming a warm conveyor belt that spans more than 10,000 kilometers. The current moves at speeds of up to 2 meters per second, carrying nearly 150 million cubic meters of water per second, a volume greater than all the world's rivers combined. This immense flow redistributes heat across the Atlantic, giving regions like the British Isles and Scandinavia far milder climates than other areas at the same latitude. But the Gulf Stream does more than warm continents. It supports an intricate network of marine life by transporting nutrients and plankton across the ocean. Many species of fish, including tuna and swordfish, migrate along the edges of this current, using its predictable flow to conserve energy during long journeys. The Gulf Stream also acts as a nursery for young marine animals. Eddies and gyres formed along its boundaries trap plankton and larval fish, creating safe pockets where life can grow. These swirling structures, some stretching hundreds of kilometers across, are biological hotspots that support feeding grounds for whales, dolphins, and seabirds. In contrast to the warm, fast-moving Gulf Stream, another type of oceanic system plays an equally important but different role: upwelling. Upwelling occurs when deep, cold, nutrient-rich water rises to the surface. This process usually happens along coastlines, where persistent winds push surface waters away, allowing water from below to replace it. Upwelling zones are among the most productive regions on Earth, accounting for nearly 50% of global marine fish catches, despite covering less than 10% of the ocean's surface. One of the most famous upwelling systems is found along the coast of Peru and Chile, driven by the Humboldt Current. This cold current flows northward along South America's western edge, bringing deep ocean nutrients into sunlight-rich waters. Here, phytoplankton blooms rapidly, forming the base of a rich food web. Sardines, anchovies, and mackerel thrive in these conditions, supporting large populations of seabirds, sea lions, and whales. The productivity of this region is so high that it influences global carbon cycles, drawing significant amounts of carbon dioxide from the atmosphere through phytoplankton growth. A similar dynamic plays out along the coast of California, where strong winds and ocean geometry create one of the most studied upwelling systems in the world. Known as the California Current System, it supports vast kelp forests, large fish populations, and migratory species such as gray whales and leatherback turtles. Scientists have monitored these waters for decades, using satellites and ocean buoys to measure changes in temperature, nutrient content, and biological activity. Their research shows that even small shifts in wind patterns can dramatically alter upwelling intensity, affecting entire ecosystems within months. Upwelling systems also play an important role in global climate regulation. When nutrient-rich waters rise to the surface, they stimulate phytoplankton blooms that absorb carbon dioxide during photosynthesis. These microscopic organisms form the foundation of the marine food chain, but their influence extends far beyond biology. When phytoplankton die, some of the carbon they contain sinks to the ocean floor, effectively removing it from the atmosphere. This biological pump is one of the most important natural mechanisms for regulating the Earth's climate, and its efficiency is closely tied to the strength of upwelling currents. Another crucial component of the ocean's pulse is the thermohaline circulation, a global conveyor belt driven by differences in water temperature and salinity. Cold, salty water is dense and sinks to the ocean floor, while warm, less salty water rises. This process creates a slow but steady movement of water that connects every major ocean basin. The thermohaline circulation is responsible for transporting oxygen to deep ocean layers and returning nutrients to the surface in certain regions. Without it, large portions of the deep sea would be deprived of life-sustaining oxygen. The interaction between wind-driven surface currents, upwelling systems, and the thermohaline circulation creates a dynamic environment in which marine life adapts and thrives. Species distribution often follows the pathways of these currents. For example, plankton blooms triggered by upwelling attract small fish, which in turn draw predators such as tuna, seals, and seabirds. Along migratory routes, large oceanic currents act like underwater highways, guiding animals along efficient paths across vast distances. Sea turtles, whales, and even certain shark species navigate these flows with remarkable precision, timing their movements to coincide with seasonal productivity. Currents also shape the physical structure of marine habitats. Coral reefs, for instance, depend on stable warm water currents to deliver food, remove waste, and maintain temperature patterns suitable for coral growth. Nutrient cycles governed by these currents influence where reefs can form and how resilient they are to stress events like bleaching. In regions where upwelling occurs too close to reef systems, the influx of cold, nutrient-rich water can disrupt coral symbiosis, but in moderate amounts, it can support nearby fisheries and enrich surrounding ecosystems. In the open ocean, drifting plankton communities illustrate the influence of currents with striking clarity. Entire assemblages of microscopic life, diatoms, copepods, and larval fish move with these flows like invisible clouds. Their presence determines where larger species will migrate to feed. Satellite imagery often captures these patterns as vast swirls of color, revealing the hidden motion of the ocean's pulse. Modern technology has greatly expanded our understanding of global currents. Satellite altimeters measure sea surface height to detect variations caused by underlying currents. Floats from the Argo program, now numbering over 4,000 across the world's oceans, drift at depth and rise periodically to transmit temperature and salinity data. These long-term datasets allow scientists to track changes in current strength, ocean heat content, and nutrient distribution. They have revealed clear signs of warming trends, shifts in current pathways, and changes in upwelling behavior, all of which influence marine ecosystems. One of the major concerns in recent years is how climate change may alter the ocean's pulse. Warmer waters can weaken major currents, including parts of the Gulf Stream system, potentially affecting weather patterns and marine productivity. Changes in wind patterns may disrupt upwelling systems, reducing the nutrient supply that sustains fisheries and wildlife. Meanwhile, The thermohaline circulation is sensitive to freshwater input from melting glaciers, which can reduce water density and slow the global conveyor belt. These shifts threaten the delicate balance that marine life depends on. Despite these challenges, the resilience of the ocean's current systems remains remarkable. They have withstood ice ages, warm periods, and natural climate oscillations for hundreds of thousands of years. Understanding their behavior offers a pathway to predicting how marine ecosystems will respond to future change. By monitoring currents, scientists can forecast shifts in fish populations, identify vulnerable habitats, and develop strategies to protect ocean life. The ocean's pulse is steady, powerful, and endlessly complex. It is the unseen force that connects distant shores, sustains vast food webs, and shapes the destiny of countless species. As the currents flow, they carry the story of life itself, one that reaches from the sunlit surface to the darkest depths. Recognizing their importance helps us appreciate the intricate systems that keep our planet alive and reminds us how vital it is to protect the ocean's delicate rhythms. In the warm, sunlit waters of tropical reefs, color becomes more than beauty. It becomes communication. Every shade, pattern, stripe, and shimmer carries meaning within this bustling underwater world. To the untrained eye, reef fish appear as simply vibrant and decorative, but scientists have uncovered a deeper truth. Their colors form a complex visual language that governs survival, courtship, territory, and even cooperation. This living symphony of color is shaped by biology, behavior, and the physics of light itself. The foundation of this language lies in specialized pigment cells known as chromatophores. These microscopic structures are embedded within the skin of many fish species. Chromatophores contain pigments that reflect, absorb, or scatter light, creating hues that range from deep blacks and reds to brilliant yellows and blues. There are several types of chromatophores. Melanophores contain dark pigments, xanthophores carry yellows, erythrophores produce reds, and iridophores reflect light using microscopic crystalline plates, creating shimmering blues and greens. By expanding or contracting these cells, fish can alter their appearance almost instantly, a remarkable adaptation that supports communication and camouflage. The ability to change color is not equal among all species. Some fish, such as parrotfish and wrasses, undergo slow but dramatic color transitions over the course of their lives. Others, including many damselfish and gobies, can shift their patterns moment to moment. One iconic example is the cuttlefish relative, the flamboyant reef octopus. But even standard reef fish display impressive control over their chromatophores. These changes are influenced by hormonal signals, environmental cues, and neural control, allowing fish to respond dynamically to threats or opportunities within their surroundings. Color also reveals important information about a fish's life stage. Many species display sexual dimorphism, meaning males and females have different colors or patterns. In some wrasse species, males transform into more vibrant versions as they mature, signaling dominance or readiness to breed. Juveniles often show distinct markings that protect them from predators or allow them to integrate into mixed species groups for safety. In some cases, the young mimic cleaner fish, tiny species that pick parasites off larger fish to reduce the likelihood of being eaten. Communication through color becomes especially apparent during mating rituals. When courting, male reef fish often intensify their colors dramatically. Fairy wrasses, for example, produce neon-like displays of blues and reds as they perform rapid swimming dances for potential mates. Male parrotfish engage in similar displays, shifting shades as they chase and circle females. These visual signals allow females to assess the fitness and health of a potential partner, as brighter colors often indicate good nutrition and strong genetics. Territory also plays a role in color communication. Many reef fish maintain carefully defended spaces on the reef, and their colors help signal their ownership. Damselfish are known for their strong territorial behavior. When threatened, they darken their bodies and display flashes of contrasting colors, signals meant to intimidate intruders. Similarly, butterfly fish use distinct patterns of stripes and eye-like spots to confuse predators and competing individuals. Studies have shown that these visual signals reduce physical confrontations by allowing fish to assess one another from a distance, avoiding unnecessary battles. One of the most fascinating aspects of color communication in reef fish is mimicry. Some species evolve to resemble others in both shape and color, gaining advantages for survival. A well-known example is the mimic octopus, but even among fish, the phenomenon is widespread. The false cleaner fish, Aspidontus taeniatus, imitates the appearance of the blue-streaked cleaner wrasse, a fish trusted by larger species to remove parasites. Rather than cleaning, the impersonator uses this trust to sneak bites of scales or mucus from unsuspecting visitors. This deceptive mimicry is so precise that even experienced predators can be fooled. Other forms of mimicry are more cooperative. Juvenile angelfish often mimic the patterns of butterfly fish to avoid aggression. Similarly, some small reef fish adopt the colors of venomous species, gaining protection by association. These intricate interactions show the evolutionary pressure that colors exert within the reef environment, where any advantage can mean the difference between life and death. Beyond mimicry, many reef fish use colors as signals of trust and cooperation. Nowhere is this more evident than in the cleaning stations scattered throughout the reef. Cleaner fish, such as the blue-streaked cleaner wrasse, wear distinctive blue and black stripes that advertise their services. Larger fish recognize these patterns and approach the cleaners with peaceful intent, often posing in ways that expose vulnerable areas for grooming. The exchange benefits both parties. Cleaners gain food, and clients remain healthy. The colors of the cleaners act as a universal code in this underwater network, reducing conflict and promoting mutualism. Color communication would be impossible without light, and the physics of how light behaves underwater adds another layer to this symphony. Water absorbs different wavelengths at different rates, causing reds and oranges to disappear quickly with depth, while blues and greens penetrate further. As a result, many reef fish have evolved to reflect or perceive wavelengths that match their environment. Some species, such as anthias and surgeonfish, display bright blues and purples that remain visible even in deeper waters. Others emphasize patterns that are more recognizable at short distances, compensating for the limited range of underwater vision. The eyes of reef fish are equally specialized. Many species can detect ultraviolet light, which humans cannot see. This UV vision allows them to communicate using patterns invisible to predators or competitors. Scientists studying damselfish have discovered that some species have UV-reflective facial markings that play a crucial role in social interactions. These hidden signals provide a private channel of communication within the chaos of the reef. Even more remarkable is the way fish perceive movement. Small shifts in color or pattern can serve as subtle cues. A flutter of fin markings can signal submission, aggression, or readiness to mate. The dynamic interplay of motion and color creates an almost continuous dialogue among reef inhabitants. In busy coral systems, where hundreds of species coexist within tight spaces, these quick exchanges help maintain order and reduce conflict. Color communication also adapts in response to environmental changes. As coral reefs experience bleaching events, their bright backgrounds can fade, altering the visual environment for fish. Some species respond by changing their colors to match the new surroundings, while others become more vulnerable to predation. Researchers have noted shifts in fish behavior following bleaching events, suggesting that the visual landscape of the reef plays a significant role in social structures and survival. Despite the beauty of reef fish colors, they come with costs. Bright patterns can attract predators, and maintaining vibrant pigments requires energy and access to certain nutrients. Yet the benefits, enhanced communication, mating success, and species recognition, outweigh these risks. This balance illustrates the delicate evolutionary trade-offs that shape the lives of reef fish. The symphony of color on tropical reefs is not a static performance. It is constantly changing. Every moment brings new interactions, a parrotfish adjusting its shades at sunrise, a cleaner wrasse signaling its intentions, or a butterflyfish flashing warning stripes as it darts through coral branches. These colors form a language older than human history, refined over millions of years of evolution. They tell stories of cooperation, competition, survival, and adaptation. Through their patterns and signals, reef fish reveal the hidden complexity of life in one of Earth's most vibrant ecosystems. As sunlight filters through the water, illuminating the coral structures below, the reef becomes a living tapestry of motion and color. Every hue carries meaning. Every pattern serves a purpose. And every fish, from the smallest goby to the stately angelfish, contributes a note to this underwater symphony. It is a world where communication transcends sound, relying instead on light, biology, and the shared rhythms of the reef. In the vastness of Earth's oceans, countless journeys unfold each day, unseen by human eyes. While we often think of migration as something carried out by whales, birds, or great herds on land, the largest daily movement of life is something far more subtle. It occurs in the darkness of the open sea, carried out by billions of tiny, drifting organisms. This immense vertical migration, performed by jellyfish, zooplankton, and microscopic animals, has been called the greatest synchronized movement on the planet. It takes place not across continents, but through water columns stretching hundreds of meters deep. This phenomenon, known as diel vertical migration, is driven by the cycle of day and night. When sunlight fades and the surface waters turn dim, multitudes of organisms rise from the deep to feed. At dawn, as light returns and predators become more active, they retreat once more into safer, darker depths. This rhythmic migration shapes the flow of energy through the ocean, influences global carbon cycles, and supports countless marine food webs. The migration involves a remarkable range of species. Among them are zooplankton, a broad category that includes tiny crustaceans called copepods, larval stages of fish and invertebrates, and gelatinous animals such as salps and small jellyfish. Even though many are no larger than a grain of rice, their collective biomass is staggering. Scientists estimate that if all zooplankton were gathered together, they would outweigh all the world's fish combined. Their daily journeys, though individually small, scale up to a planetary event. The reasons behind vertical migration are rooted in survival. Near the surface, sunlight supports the growth of phytoplankton, microscopic plants that form the foundation of marine food chains. These plant-like organisms release oxygen, capture sunlight, and produce nearly half of the world's atmospheric oxygen. They also form the primary food source for many zooplankton. But the surface is also where predators hunt most effectively, using sunlight to detect movement and silhouettes. By ascending under the cover of night, zooplankton feed when danger is minimized. As dawn approaches, the increasing brightness signals a retreat. Some species descend hundreds of meters within a short period, moving down water faster than their small bodies would suggest possible. Their migrations follow a strict rhythm, governed by light levels, internal biological clocks, and chemical cues in the water. Jellyfish and gelatinous zooplankton also join this nightly ascent. Though often seen as slow or passive drifters, many jellyfish species are surprisingly active. Some pulsate upward, with coordinated contractions of their bells, while others rely on buoyancy shifts controlled by the arrangement of ions within their tissues. In colder, deeper waters, species like the Paraphila, the crowned jellyfish, perform their own vertical migrations, rising at night to feed on planktonic crustaceans before retreating to the deep. These movements have far-reaching ecological consequences. As zooplankton travel upward and downward, they transport organic material through the ocean. When they feed on phytoplankton near the surface, they convert that energy into their own biomass. Later, as they descend, they release waste products, tiny pellets known as marine snow, that sink into deeper waters. This transport process, known as the biological carbon pump, plays a crucial role in regulating Earth's climate. It moves carbon from the surface into the deep ocean, where it can remain trapped for centuries or even millennia. Recent studies estimate that this giant daily migration helps sequester over 2 billion tons of carbon each year. Without it, atmospheric carbon dioxide levels would be far higher, shifting the planet's climate in significant ways. This hidden cycle reminds us that even the smallest organisms can shape global systems on staggering scales. For decades, scientists struggled to measure this migration. It was simply too vast and too deep. Early sonar systems developed during World War II revealed something unexpected, a mysterious layer in the ocean that rose and fell each day, confounding submarine crews. This deep scattering layer turned out to be composed of millions of migrating organisms whose tiny bodies reflected sonar pulses. What began as a wartime curiosity eventually became a key discovery in marine biology. In recent years, satellite observations have expanded our understanding of these migrations. While satellites cannot directly see animals in deep water, they can measure factors that influence their movement, such as surface productivity and light penetration. High-resolution ocean color satellites reveal where phytoplankton blooms occur, helping scientists predict where large zooplankton migrations are likely to be most intense. Some instruments detect subtle changes in water properties associated with the nightly rise of planktonic organisms. New technologies, including autonomous underwater vehicles and deep-sea profiling instruments, have provided even more insight. These devices travel through water columns, recording sound scattering, temperature, and particle density. Using their data, researchers have confirmed that DL vertical migration occurs not only in tropical waters, but in nearly every ocean on Earth, from polar seas, where darkness lasts for months, to the equatorial Pacific, where light cycles remain constant year-round. Perhaps most astonishing is the scale of coordination involved. Unlike birds or mammals, zooplankton do not communicate or travel in organized groups. Their migration is driven by instinct and environmental cues, yet billions ascend and descend almost simultaneously. This synchronized behavior has been compared to a silent underwater tide, one not composed of water, but of life. Even fish participate in these vertical migrations. Many species of lanternfish, which glow with bioluminescent organs, spend their days hundreds of meters below the surface. At night, they rise to hunt smaller zooplankton, forming part of a chain of movements that cascades upward through the food web. These nightly hunts shape the distribution of predators and prey throughout the open ocean, influencing everything from the feeding patterns of whales to the location of commercial fisheries. The migration also plays a role in spreading nutrients across ocean layers. As organisms travel, they mix water masses in subtle ways, blending oxygen, trace minerals, and organic matter. Though each tiny movement seems insignificant, the collective effect contributes to the slow heartbeat of the ocean, stirring waters that might otherwise remain still for decades. Climate change poses new challenges to this delicate process. Rising temperatures affect the distribution of phytoplankton, altering where food is available for migrating zooplankton. Ocean acidification impacts the ability of some species, particularly those with calcium-based shells, to survive and grow. Changes in predator populations, such as fish stocks altered by overfishing, can disrupt the timing and scale of migrations. Even small shifts in these patterns could have wide-reaching consequences for marine ecosystems and the global carbon cycle. Despite these pressures, deal vertical migration remains one of the most resilient and ancient behaviors in the ocean. Fossil evidence suggests that similar migrations occurred hundreds of millions of years ago, long before the rise of complex vertebrates. This behavior has survived mass extinctions, ice ages, and the shifting geography of continents. It represents one of nature's most enduring rhythms, a nightly journey repeated across generations without pause. As the sun sets over the ocean, this vast migration begins anew. Jellyfish drift upward from quiet depths. Copepods rise in dense clouds, moving with the fading of the light. Lanternfish follow behind, tracing glowing arcs in the water. Kilometers above them, satellites orbit silently, unaware that below, the largest movement of life on Earth is unfolding in perfect synchrony. Though invisible to us, this migration is a reminder of the unseen forces that shape our world. Countless drifting organisms, fragile, translucent, and small, collectively influence the chemistry of the oceans, the balance of the atmosphere, and the cycles that keep Earth alive. Their nightly journey, simple yet profound, connects every level of the marine ecosystem, linking surface sunlight to deep-sea darkness. In this hidden world of drifting multitudes, nature reveals its quiet power. The greatest migration on the planet is not one of great distance, but of depth. A rhythmic rise and fall that continues, night after night, across all the oceans of the Earth. In the shadowed world beneath the waves, few creatures inspire as much fascination as the octopus. With its fluid movements, shifting colors, and extraordinary intelligence, it feels almost otherworldly. Scientists have long considered the octopus one of the most remarkable animals on the planet, not for its size or strength, but for its mind. Among all invertebrates, it stands alone, capable of problem-solving, exploration, adaptation, and a kind of creativity that challenges our understanding of marine life. Part of what makes the octopus so enigmatic is its anatomy. Unlike fish, mammals, or reptiles, an octopus has no bones and no rigid structure. Its soft, flexible body allows it to squeeze through openings no larger than its beak, a hard, parrot-like structure that is the only solid part of its form. This ability to flow through cracks and crevices offers both protection and opportunity, giving the octopus access to hiding spots and hunting grounds unavailable to most predators. Perhaps the most famous ability of the octopus is its camouflage. It is widely considered the most skilled camouflage artist in the animal kingdom. Using specialized skin cells, chromatophores, iridophores, and leucophores, an octopus can change its color, brightness, pattern, and even the texture of its skin in less than a second. Chromatophores expand and contract to display pigments, iridophores reflect light to create iridescence, and leucophores scatter ambient light to mimic surrounding colors. Working together, these cells allow the octopus to match coral, sand, rock, or shadow with astonishing precision. What makes this even more remarkable is that octopuses are colorblind. They cannot see hues the way humans do. Instead, Scientists believe they perceive changes in light, contrast, and polarization, which they use to control their living camouflage system. Some studies suggest that their skin may contain light-sensitive proteins similar to those in their eyes, meaning the skin itself might see enough to adjust its appearance. This camouflage is more than a visual trick. It is a sophisticated survival strategy. Many octopus species live just a few years, and their soft bodies make them vulnerable to predators like sharks, groupers, and seals. To survive, they rely on deception. They blend into backgrounds, mimic seaweed or stone, and even impersonate other animals. The Indonesian mimic octopus can imitate up to 15 species, including venomous lionfish and flatfish, adjusting its posture and movement to match their appearance. This form of mimicry is rare among invertebrates and underscores the octopus's adaptability. Beyond camouflage, the octopus demonstrates intelligence in ways few marine animals can. Researchers have documented octopuses solving puzzles, navigating mazes, and manipulating objects with surprising dexterity. Their arms contain more than 2/3 of their neurons, allowing each arm to explore and react almost independently. This distributed intelligence means an octopus can coordinate 8 limbs while simultaneously processing information from its surroundings. Studies conducted in marine laboratories have shown octopuses unscrewing jar lids to reach food inside, lifting levers, and remembering solutions to tasks for weeks. Individual octopuses exhibit distinct personalities, some bold, some cautious, some curious, others more reserved. This behavioral diversity is unusual in invertebrates and has intrigued scientists trying to understand the roots of animal cognition. One of the most well-known examples of octopus problem-solving occurred at the University of Otago in New Zealand, where an octopus named Inky gained global fame. Inky escaped from his enclosure, crossed the laboratory floor, found a drain pipe, and slid through it to reach the ocean. This escape demonstrated not only physical flexibility, but spatial awareness, exploration, and decision-making. Skills rarely attributed to animals without backbones. Observations in the wild support the idea of octopus intelligence. In some regions, particularly off Australia, octopuses have been seen collecting discarded coconut shells or seashells, carrying them across the sea floor and assembling them into shelters when needed. This behavior suggests foresight, preparation, and tool use. Only a small number of species on Earth, humans, primates, certain birds, and now octopuses, regularly use tools in this way. Their brains, though very different from ours, are large relative to their body size. The octopus nervous system includes approximately 500 million neurons, comparable to that of some small mammals. Yet their brain structure diverged from the vertebrate design more than 500 million years ago. This makes octopuses an extraordinary example of convergent evolution, where intelligence evolves independently in unrelated lineages. Their cognitive abilities challenge assumptions about what is necessary for complex thinking. The octopus's short lifespan also adds to the mystery. Most species live only 1 to 3 years. After mating, females guard their eggs with extreme devotion, often refusing food during the incubation period. They wash and oxygenate the eggs constantly until they hatch. Shortly after, the female dies, exhausted from her vigil. Males also die soon after reproduction. Given this brief life, scientists wonder how octopuses develop such sophisticated intelligence in such a short time. Their rapid learning and curiosity hint at a life lived intensely, with each moment spent exploring, adapting, and interacting with their environment. Communication is another surprising aspect of octopus behavior. Though generally solitary, they express themselves through subtle changes in posture, color, and texture. A sudden shift to deep red might signal agitation, while pale or mottled patterns may show relaxation or submission. These signals are not as structured as those of social animals, but they reveal emotional responses and decision-making processes. In several studies, octopuses reacted differently to different human handlers, showing signs of recognition, preference, or avoidance. Even their hunting techniques display innovation. Some species stalk prey quietly, using camouflage to approach unnoticed. Others employ ambush hunting, spreading their arms over coral crevices to trap small fish or crustaceans. A few species use jet propulsion to launch themselves rapidly forward, startling prey into motion. Octopuses have even been observed throwing debris, sand, shells, or algae toward objects or animals, a behavior that may serve as distraction or territorial defense. It is one of the few examples of projectile use in a marine invertebrate. Despite their intelligence, octopuses remain elusive. Their bodies leave few fossils, and their soft tissues rarely preserve in geological records. Much of what we know comes from modern observation and experimentation. New species continue to be discovered, especially in deeper waters where octopus diversity is still poorly understood. Submersible missions have revealed species like the Dumbo octopus, with ear-like fins, living thousands of meters below the surface. These deep-sea octopuses often grow slowly, withstand immense pressure, and live far longer than their shallow water relatives, offering scientists new perspectives on octopus evolution. Octopus research has expanded rapidly in recent decades as scientists seek to understand how intelligence emerges in such a unique organism. Genome sequencing has revealed that octopuses possess an unusually large number of genes associated with neural development and adaptability. They also exhibit high levels of RNA editing, a process that allows them to modify their own genetic instructions at the cellular level. This may explain their extraordinary sensory abilities and rapid responses to environmental changes. Conservation concerns add urgency to this research. Climate change, pollution, and habitat degradation affect octopus populations, particularly those in coral reef habitats or shallow coastal waters. Rising temperatures can disrupt breeding cycles, while ocean acidification affects prey availability. Although many octopus species reproduce quickly and adapt well to changing environments, others, especially deep-sea species, may be more vulnerable. Yet, despite these challenges, the octopus remains a symbol of resilience and ingenuity. Its intelligence evolved in isolation, guided by pressures very different from those shaping vertebrate minds. Its camouflage is a masterpiece of biological engineering, its behavior a testament to the complexity that can arise in unexpected places. As we watch an octopus glide across the seafloor, shifting from pebble to shadow to sand, we witness the artistry of a creature shaped by millions of years of adaptation. Hidden within its fluid form is a mind capable of exploration, invention, and transformation. In the quiet depths of the ocean, the octopus continues to surprise us. An ancient being, a master of disappearance, and one of the most remarkable intelligences of the deep. Beneath the sunlit surface of the ocean lies a world almost entirely alien to human experience. Beyond 1,000 meters, sunlight fades to nothing, pressure rises to levels that would crush unprotected bodies, and temperatures drop near freezing. For centuries, this deep-sea realm remained invisible, a silent abyss where life was thought to be sparse and fragile. It was not until the advent of modern submersibles that scientists could venture into these depths and witness the extraordinary ecosystems that thrive there. Among the pioneering vessels, Alvin stands out as one of the most influential in the history of oceanography. Launched in 1964 by the Woods Hole Oceanographic Institution, Alvin was designed to carry a crew of 2 scientists and 1 pilot to depths previously unreachable. Its initial purpose was broad oceanographic exploration, but the submersible quickly became famous for its deep-sea discoveries, transforming our understanding of life in the abyss. Alvin is a compact, pressure-resistant craft with thick steel hulls and acrylic viewports, equipped with manipulator arms, lights, and sampling tools. Its ability to navigate, hover, and document life in complete darkness allowed scientists to explore environments long assumed to be barren. The first major revelation came in 1977, during dives along the Galapagos Rift in the Pacific Ocean. Alvin's team discovered hydrothermal vents, or black smokers, spewing superheated water rich in minerals like iron, copper, and sulfides. Surrounding these vents was a community of organisms entirely unknown to science. Giant tube worms, clams, mussels, and shrimp clustered around the vent chimneys, thriving in waters over 350 degrees Celsius near the vents, despite near-freezing temperatures just meters away. This ecosystem was powered not by sunlight, as in surface food chains, but by chemosynthesis, a process where bacteria convert chemicals from vent emissions into organic energy. The discovery challenged fundamental assumptions about the conditions necessary for life. Alvin's expeditions revealed remarkable species firsthand. Riftia pachyptilla, the giant tubeworm, can grow over 2 meters long and host symbiotic bacteria in a specialized organ called a trophosome. These bacteria process hydrogen sulfide, allowing the worm to survive without sunlight or traditional food. Alvin's manipulator arms collected specimens carefully, enabling laboratory studies that confirmed this extraordinary biological partnership. Subsequent dives found entire vent communities teeming with life: alvinellid worms, which tolerate extreme heat and acidity, vent crabs that scrape bacteria off surfaces, and blind shrimp whose senses are finely tuned to detect chemical cues from the vents. Alvin also documented an array of bioluminescent fish and invertebrates, adaptations critical for survival in total darkness. Many deep-sea species emit light through specialized organs called photophores. These lights serve multiple purposes: attracting prey, communicating, camouflaging against predators, or confusing attackers. Among the bioluminescent fish observed, species of anglerfish captivated scientists with their unique hunting adaptation. Female anglerfish possess a luminous lure extending from their heads, which they wiggle to attract smaller fish in pitch-black waters. Alvin's cameras recorded interactions between predator and prey, revealing behaviors that were entirely invisible before direct observation. In addition to known species, Alvin has brought countless new species to light. Each dive reveals previously undocumented forms of life, often with extraordinary adaptations. Deep-sea amphipods the size of small lobsters, transparent jellyfish with pulsating blue glows, and flatfish that appear almost invisible against dark sediment, all exemplify the diversity of the abyss. Some of these species occupy niches that have no analog in shallower ecosystems, exploiting chemical, thermal, or pressure extremes inaccessible elsewhere. The discovery of these organisms has significantly expanded biological classification, revealing evolutionary paths shaped by conditions unimaginable to surface-dwelling animals. The technological innovations of Alvin also allowed for detailed mapping of the seafloor. Using sonar and onboard cameras, researchers identified hydrothermal vent fields, underwater mountain ranges, and tectonic rift zones, linking biological discoveries to geological processes. Vent communities were found along mid-ocean ridges spanning thousands of kilometers, confirming that chemosynthetic ecosystems are widespread, though scattered across the globe. Alvin dives demonstrated that these habitats are fragile and highly localized. Even minor disruptions from volcanic activity or sediment movement can wipe out entire colonies, highlighting the vulnerability of deep-sea life. Life at these depths requires extraordinary physiological adaptations. Deep-sea fish and invertebrates must withstand pressures exceeding 400 atmospheres at 4,000 meters. Their cell membranes and proteins are specially structured to function under intense compression, and many possess reduced skeletal structures or fluid-filled tissues to resist crushing. Bioluminescence and specialized eyes allow them to navigate and hunt in near-total darkness. Alvin's observations of feeding, mating, and sheltering behaviors have provided insight into these extreme adaptations, revealing a world that is both alien and highly dynamic. Beyond hydrothermal vents, Alvin has explored cold seeps, methane-rich environments where chemosynthetic bacteria again support complex communities. Clams, mussels, and tube worms inhabit these sites, feeding on microbial partners in a strikingly similar fashion to vent communities. Such ecosystems demonstrate the versatility of life in harnessing chemical energy from the Earth itself, independent of sunlight. Alvin's sampling of these sites has helped scientists understand how deep-sea organisms respond to temperature, chemical gradients, and microhabitats, providing models for studying life's limits. Alvin's deep-sea expeditions have also revealed unexpected behaviors. Deep-sea octopuses, for instance, have been observed using discarded shells as shelters. Deep-sea crabs and shrimp form clusters around vent chimneys, creating local food webs that operate on remarkably short cycles compared to the slow processes in surrounding sediment. By filming and sampling these interactions, Alvin has allowed scientists to piece together food chains and ecological networks in a region where conventional observation is impossible. Another remarkable discovery is the diversity of life around submarine volcanoes. Alvin dives near the East Pacific Rise, and other active sites revealed new species of anemones, echinoderms, and small fish that thrive in unstable conditions. These ecosystems endure frequent eruptions, fluctuating temperatures, and chemical shifts, yet they persist, suggesting a resilience shaped by evolutionary pressure and isolation. Alvin's exploration has helped catalog these species and monitor how communities respond over time, providing essential baseline data for conservation efforts. Alvin's contributions extend beyond biology. It has transformed how humans understand the oceans and their role in Earth's systems. Hydrothermal vents release minerals and heat that influence ocean chemistry, including nutrient availability for plankton at greater depths. Bioluminescent species play roles in carbon cycling and predator-prey dynamics. The knowledge gained from Alvin dives informs models of ocean productivity, deep-sea ecology, and even the search for extraterrestrial life, where hydrothermal vents serve as analogues for environments on moons like Europa or Enceladus. Over its decades of service, Alvin has descended more than 4,000 times, reaching depths over 4,500 meters. Its expeditions have led to the description of hundreds of new species and redefined the scientific understanding of life under extreme conditions. Every dive demonstrates that the deep sea is not a barren wasteland, but a world of astonishing diversity where life thrives in the dark, in ways both alien and intimately connected to Earth's geological and chemical cycles. In the darkness of the abyss, Alvin has revealed creatures that seem almost fantastical. Glowing fish, ghostly jellyfish, giant tube worms, and cunning crustaceans. These discoveries illuminate not only the resilience of life, but its boundless capacity for adaptation. Each observation reminds us that the oceans hold countless secrets, many of them critical for understanding our planet, our climate, and the limits of life itself. Alvin's legacy is a testament to human curiosity and ingenuity, a journey into the black abyss that continues to inspire wonder, discovery, and reverence for the unseen worlds beneath the waves. Thank you for joining us on this journey through the hidden wonders of the ocean. We hope this documentary has brought you a moment of peace and a deeper connection to the natural world. From the gentle sway of coral reefs to the mysterious depths of the abyss, the ocean's rhythms can calm the mind and soothe the soul. If you enjoyed this video, don't forget to subscribe to Deep Sleep Documentaries. Subscribing makes it easy to find these calming stories whenever you need a quiet moment. And if this documentary helped you relax, a simple like can help others discover it too. Your support means the world to us. We'll see you again in the next relaxing exploration of nature's wonders. Until then, take a deep breath, let the calm settle in, and allow the ocean's serenity to linger with you. Until the next time, I wish you a good sleep.